Medical Biology for first year dentistry students, lecture 2 and 3, prepared by assistant lecturer Hajil Kazam Turkey. The cell structure. In general, all cells consist of three main parts, which are the cell membrane, or which is also called the plasma membrane. Second, the cell organelles, and third, the matrix. The cell membrane or the plasma membrane. The membrane that surrounds all parts of the cell. It separates the internal environment of the cell from the external environment. Also, it regulates the entrance and exit of molecules into the cell. Structure of the plasma membrane. Plasma membrane structure consists of a fluid phospholipid bilayer in which embedded proteins form a mosaic pattern. This is called the fluid mosaic model. As we said, plasma membrane is a phospholipid bilayer with embedded proteins. The phospholipid molecules are arranged as the following. First, the hydrophilic polar heads, which is a water-loving, face the surface of the cell where water is found. The second is hydrophobic nonpolar cells, which is called water-fearing. This is face each other. Because they are bilayer, each layer consists of hydrophilic and hydrophobic, so the hydrophobic nonpolar cells face each other, while the hydrophobic polars face the outer environment from one side and the inner parts of the cell from the other side. Cholesterol is another lipid found in animal plasma membrane, and steroids are found in plant plasma membrane. The proteins. Proteins in plasma membrane may be peripheral or integral. Peripheral proteins on the inside surface are held by skeletal filaments, while the integral proteins are embedded in the membrane. The carbohydrate chains occur only on the outside surface of the plasma membrane and involved in marking the cell. They are found in two types: glycolipids, which consist of phospholipid plus carbohydrates, mostly sugar, and second, the glycoproteins, which consists of proteins plus carbohydrates, also sugar. In this picture, we can see the plasma membrane structure. The phospholipid bilayer with red color. These two layers, each one consists of hydrophilic and hydrophobic parts. The round is, or the spherical part, is the hydrophilic, and the tail is the hydrophobic, which face each other in two bilayer. And we can see also the proteins, peripheral proteins and integral proteins with the blue color. The green color for carbohydrates, two types of carbohydrates. With sugar that binds to protein is called the glycoprotein, while the sugar which binds to lipids is called the glycolipids, and each are outside of the plasma membrane. Functions of plasma membrane. Plasma membrane have many functions, include: first, communication by receptors that recognize and respond to signals. Second, the intracellular connection. Establish a flexible boundary, protect cellular content, and support cell structure. Third, physical barrier. Phospholipid bilayer separates substances inside and outside the cell. Fourth, plasma membrane selective permeability regulates entry and exit of different substances, allows some molecules in, others are kept out. Permeability of plasma membrane. The plasma membrane is differentially permeable. This means that some substances can move across the membrane and some cannot. The ways of crossing a plasma membrane are passive transport and active transport. First, the passive transport. In this method, There is no use for chemical energy because the molecules are moving down their concentration gradient. Passive transport includes diffusion and facilitated transport. In diffusion, there is no need for a carrier protein. This is the process in which the movement of molecules is from higher to lower concentration until equilibrium is achieved and molecules distributed equally. And for example, the gas exchange in lung during respiration. Osmosis. Osmosis is the process of diffusion of water across a differentially permeable membrane due to concentration differences. There are two types of solutions according to the osmotic pressure: isotonic solution, hypotonic solution, and hypertonic solution. In isotonic solution, no net gain or loss of water, no change in cell, concentration of solute and concentration of water inside the cell equal to the concentration of solute and concentration of water outside the cell. So if a cell is placed in an isotonic solution, there is no change. Second, the hypotonic solution, the solution that causes the cell to swell or even to burst, which is called the cytolysis. A solution with lower concentration of solute, this means higher concentration of water in the same solution than inside the cell. So if a cell is placed in hypotonic solution, in this solution, water enters the cell and the net movement of water is from outside to inside, and this leading to swelling. In hypertonic solution, the solution that causes the cell to shrink or to shrivel, which is called the plasmolysis. A solution with higher concentration of solute, which means the lower concentration of water than inside the cell. So if a cell is placed in hypertonic solution, water leaves the cell, the net movement of water is from inside to outside of the cell, and this is called plasmolysis or shrinkage of the cell. In these two pictures, we can see the process of cytolysis. This is the swelling of cells in hypotonic solution, and the right picture is the process of plasmolysis, which is the shrinkage of cells when it puts in a hypertonic solution. The second form of passive transport is facilitated transport. In this method, the passage of molecules such as glucose and amino acids across the plasma membrane. There is no need for chemical energy, but a need for a specific carrier protein. During facilitated transport, a carrier protein speeds the rate at which the solute crosses the plasma membrane toward a lower concentration. The carrier protein undergoes a change in shape as it moves a solute across the membrane. The second type of transport across the plasma membrane is active transport. In this method, molecules are transported from low concentration to high concentration against their concentration gradient. So this requires a carrier protein plus chemical energy represented by adenosine triphosphate ATP. Proteins in active transport are called pumps. The sodium-potassium pump is an active pump in all animal cells associated with nerve and muscle cells. Moves the sodium to the outside and the potassium to the inside of the cell. This picture shows how sodium-potassium pump works. First, the carrier protein with blue color has a shape that allows it to take the sodium particle. Second, the ATP is split into ADP and phosphate group, and is attached to the carrier. The change in shape results in release of sodium from inside to outside. The carrier has now the shape that allows the potassium to enter. Phosphate group is released from the carrier, and the potassium particles released to the inside of the cell. By this transport method of sodium and potassium molecules, the nerve impulse or the neuromuscular order transported from neuron to neuron or from neuron to muscular fibers respectively. Membrane-associated transport. This method used to carry molecules, macromolecules, large molecules such as polysaccharides or polypeptides, which are too large to be transported by carrier proteins. The molecules are transported by vesicle formation, and this method considered as form of active transport because they need chemical energy to do. There are two types of membrane-associated transport or two methods, which are the exocytosis and endocytosis. Exocytosis. Vesicles have been produced by Golgi apparatus and contain proteins. Exocytosis transports secretions of cells to the outside, from inside of the cell to the outside. An example of the transported substances, the hormones or the enzymes. Endocytosis. Vesicles originated as invaginations from the plasma membrane, which envelop the substances, and the membrane pinch off to form the intracellular vesicle. Endocytosis transports substances from outside to inside of the cell, and there are three ways for endocytosis, which are phagocytosis, pinocytosis, and receptor-mediated endocytosis. This picture shows the three methods of endocytosis. First picture, phagocytosis. The process of endocytosis when the material taken in is large, such as food particle or another cell. It's common in unicellular organisms such as amoeba. For example, human white blood cells that engulf elements by the process of phagocytosis. The second is pinocytosis, which is the process of endocytosis when the material is liquid or very small particle. For example, cells that line the kidney tubules, and this helps in filtration of human waste. Third is receptor-mediated endocytosis, form of pinocytosis that is specific because it uses a receptor protein that is shaped as a specific for molecule, such as a vitamin. The receptors are found at one location in the plasma membrane and called coated pits. This process is selective and much more efficient than ordinary pinocytosis. Junctions between cells. There are three types of junctions between cells, which are adhesion junctions, tight junctions, and gap junctions. Here we have the pictures for the three types of junctions. First, adhesion junctions. Cells are joined to each other by intracellular filaments. You can see the filaments. The result is a sturdy but flexible sheet of cells. This is located in stretched tissue cells. For example, the heart cell and the bladder cells. The second is tight junction, when the plasma tissue proteins attach to each other, producing a zipper-like fastening. This is located in tissue that serve as barriers. For example, urine stays within the kidney's tubules. Third, the gap junction, when two identical plasma membrane channels join to permit small molecules and ions pass between the two cells. For example, smooth muscle cells. The extracellular matrix, a meshwork of polysaccharides and proteins that's produced from the cell itself. It supports the cells and also affects its behavior. It's either flexible like in cartilage or solid like in bone. Thank you for listening.